Malaria parasites are widespread among chimpanzees and gorillas, but for the first time, they've been detected in wild bonobos, a chimp cousin. Researchers discovered the parasites after analyzing ape fecal matter gathered from the forest floor. The findings could help inform further research into how the parasite is transmitted across species, leading to malaria disease, which killed 438,000 people in 2015. Previous studies may have missed infected bonobo, paniscus, populations, so researchers at the University of Pennsylvania conducted a more extensive study. The researchers increased the number and places they sampled wild bonobo populations. Wild bonobos are found in the forests of Central Africa and south of the Congo River in the Democratic Republic of the Congo, DRC. Although the researchers found evidence of a new malaria species in bonobos, it was limited to the easternmost part of the bonobo range. The research team was led by Dr Beatrice Hahn, a professor of microbiology in the Perelman School of Medicine at the University of Pennsylvania. Dr. Hahn's lab studies 8 relatives of human pathogens such as HIV and malaria, to learn more about the microbes that cause these diseases in humans. But African non-human primates are highly endangered and require non-invasive sampling techniques to learn more about their health. This is why Dr. Hahn's team developed a sensitive test that allows them to obtain genetic information of malaria parasites from ape fecal matter. Not finding any evidence of malaria in wild bonobos just didn't make sense, given that captive bonobos are susceptible to this infection, Dr. Hahn said. According to the World Malaria Report, there were 214 million cases of malaria globally in 2015 and 438,000 deaths from the disease, down 37% and 60%, respectively, since 2000. Malaria is caused by 4 kinds of parasites in humans, but Plasmodium falciparum is the most prevalent and lethal of the malaria parasites. Usually, people become infected by being bitten by an infected female Anopheles mosquito. Only Anopheles mosquitoes can transmit malaria, and they must have been infected through a previous blood meal taken from an infected person. Symptoms of malaria include fever and flu-like illness, including shaking, chills, headache, muscle aches, and tiredness. Nausea, vomiting, and diarrhea may also occur.It can also cause anemia and jaundice, yellow coloring of the skin and eyes, because of the loss of red blood cells. If not treated rapidly, the infection can become severe and may cause kidney failure, seizures, mental confusion, coma, and death. More work needs to be done to combat the parasite, especially since the barriers that prevent cross-species transmission are not well understood. This photomicrograph of a blood smear contains a macro and micrometacyte of the Plasmodium falciparum parasite. Within a few minutes after the Anopheles mosquito ingests the metacytes, micrometacytes develop into microgametes, which are able to fertilize gametes. We look for biological loopholes to potentially exploit the life history of these pathogens to better understand how they cross over to humans. Dr. Hahn's team found that bonobos are susceptible to a wide variety of Plasmodium malaria parasites, including a previously unknown Lavarania species that is specific to bonobos. These Lavarania parasites are close relatives of the human malaria parasite Plasmodium falciparum, the most prevalent and lethal of the malaria parasites that infect people. It seems likely that these parasites co-evolved with African apes, suggesting that the ancestors of bonobos were infected, and implying that most wild living communities of bonobos have somehow lost their malaria parasites, said co-author Paul Sharp, Ph.D., an evolutionary biologist from the University of Edinburgh. The researchers tested 1,556 fecal samples from 11 field sites, and identified a high prevalence of Lavarania infections in an area called Lualaba, a remote region in the Eastern Democratic Republic of the Congo. Until recently, there were 6 known ape Lavarania species that were specifically associated with a single host species in wild populations, 3 in chimpanzees and 3 in western gorillas. In 2010, Dr. Hahn and colleagues discovered that gorillas were the origin of the human malaria parasite Plasmodium falciparum. One surprising finding from the new study was that the bonobos harbor Plasmodium gaboni, which was previously only found in chimpanzees, as well as a new Lavarania species called P. lamamiensis, in recognition of the recently established Lamami National Park.The research team tried to understand why malaria parasites were absent from most other bonobo field sites. However, neither parasite seasonality nor bonobo population structure could explain what they observed. For now, the geographic restriction of bonobo Plasmodium infection remains a mystery, said co-first author Weimin Liu, a senior research investigator in Dr. Hahn's lab. We have yet to identify the causes, said co-first author Scott Sherrill Minks, a Hahn Lab postdoctoral fellow. We looked at what plants bonobos eat, and what types of bacteria make up their gut microbiome, but these could not explain the absence of Plasmodium from most of the bonobo sites. From this, we suspect that factors that influence parasite transmission are involved. As researchers consider how malaria can be eliminated from the human population, Dr. Hahn notes that it is important to understand more about these ape parasites, what factors affect their distribution and host specificity, and whether there are circumstances under which any of them could again jump into humans. Dr. Hahn is experienced with this area of research. In 2006, her lab and collaborators confirmed that the virus responsible for the AIDS pandemic in humans, HIV, is of chimpanzee origin.